So how can we use SoyBase genetic map displays to identify potential candidate genes for a GWAS QTL? Well, it's pretty simple. Pictured here is the genetic display from D1 alpha linkage group, and on the right hand side we have the map, which is the soybean GM composite 2003 genetic map as we're used to seeing it. And then on the other side is what we call soybean GM GWAS. And what you're seeing here is GWAS QTLs, which we've mined from the literature and placed on the sequence map, which is viewed right here. And the linkages between the sequence map and the genetic map are shown with connecting blue lines, and they are essentially SNPs and other sequence-based markers that could be placed on the physical map of soybean in terms of base pairs and not centimorgans in distance. If we look for this group of QTLs, which are for seed color, we see that they came from one paper, and they all appear at this resolution anyway to stack up, which probably means they're closely linked SNPs. So let's see if we can find candidate genes for those three SNPs. Well, first we need to zoom in to the genetic map, and we do that simply by determining about where the position of those GWAS QTLs are, and then using our arrows to snip off the genetic map that we don't want to look at. And so if we zoom into this area, we see that those three GWAS QTLs that look like they were stacked one on top of the other are actually not, but they're closely positioned to one another. And in fact, they look like they might be linked to this gene, and or maybe this gene. So how can we take this potential list of candidate genes and turn it into a real live, really good candidate gene? Well, one way is to click on each one of these gene models in succession and look at what SoyBase has to say about the functional annotation and the tissues in which those genes are expressed. Being that this is a seed coat color gene, we'd expect to see expression at least in the seeds, if not specifically in the seed coat. So what we'll do is we'll step through looking at each one of these genes independently. So if we look at this gene, we'll see that SoyBase has annotations for this gene, and some of the gene annotations are listed here. And as we can see, it looks like it's a protein phosphorylation gene. It might be involved in a transmembrane receptor, but nothing here screams, you know, I may be involved in seed coat color. Then if we look down here at the expression patterns, we see that in seeds, there's practically no expression of this gene at any stage of development that was captured in this experiment. So let's look at the next gene in the list, and from the annotations we see that it's probably an acetyltransferase. There's nothing in here that screams I'm involved in the seed coat whatsoever. And if we look down here at the expression patterns, we see that only is this gene involved in maybe a mild bit of expression very early in seeds in development. So if we look at the third gene and we look up here at the annotations, first thing our eye is drawn to is this Arabidopsis trichome bifurengence like 27 gene, and it's involved in trichome bifurengence, which makes you think that, well, maybe it's got something to do with color. It also may be xyloglucan metabolism. It also may be involved in cell wall organization, which is really interesting, and it may be an integral component of the membrane, and that's very interesting. And if we look down here at our expression patterns and tissues, we see that in fact this one is very highly expressed in seeds, which is what we would expect to find for a gene involved in seed coat color. Now, SoyBase has many more expression experiments than what you see here, and to see those, all you have to do is come down and click the here, and you'll be taken to our Expression Explorer page. And here are the two experiments that we saw earlier. and we see that RNA-seq captures only one experiment captured that gene. And if we look down here at the soy chip experiments, we see that that gene was not represented on the soy chip. That's because it's a assembly 2 gene, and they were not around when the soy chip was developed. However, we can convert assembly 2 gene into assembly 1 gene name simply by coming down here and hitting the submit button. And then we see that this gene is analogous to GLIMA 01G04100. And if we copy that and then go to any one of our experiment pages and simply replace that GLIMA ID with this one, and then we can see that there's an awful lot more genes that we can get, or a lot more experiments that we can get information for this gene. And if we start looking at these in depth, we see that here's an experiment of the cultivar Williams where they looked at cotyledon stage seeds, and there's 7 different stages of cotyledon development they looked at, and we can see that there seems to be expression in the cotyledon stage in Williams, certainly more than it was in 1 and certainly more than it was at stage 7. We look at the next experiment, which is Williams 43 seeds at different stages of development, all the way up to whole seeds at 22 to 24 days after flowering. And if we peruse the values, we can see that 12 days after flowering, whole seed has a fairly high expression. Whole seed here has a fairly high expression, but also the 400 to 500 milligram seeds also have comparatively high expression. And if we look down at this next large experiment and we peruse the values, we can see that the values start to come really up in Williams 82 early maturation stage embryonic axis stele and the embryonic cotyledon, abaxial parenchyma and embryonic adaxial epidermis, which shows through partially through the seed coat, so they might be involved or show us that this gene is involved in seed coat color. And if we peruse more of the experiments, here's an experiment with Williams 82 at the globular stage of development of seeds. And we peruse this and we see, in fact, the highest expression is in the seed coat outer integument. And if we look down a little further, we can see that at the heart stage in Williams 82, seed coat outer integument again was the tissue that had the most expression of this gene. And then we look at this again at Williams 82. We got cotyledon stage seed coat outer integument again had the highest, one of the highest expressions. So thus we can probably conclude that this gene is certainly expressed in seed and certainly expressed in the integument of the seed coat. Thus, of all the genes we surveyed, this is the most likely to be involved in seed coat color by virtue of its expression pattern and its functional annotations. This has been a SoyBase production. For more information about SoyBase, go to www.soybase.org.